Should I stop the song?
Yeah, wait. Sucks or copy? It's all
Yes,
fake.
sir.
We are on the
Yes,
go.
sir.
Please stop.
Yes.
Stop the music and Succor, it's all yours.
Sir, we are having Preeta with us.
Yeah, Succor, please start.
Sure. Good afternoon, everybody, and a very good morning to our speaker, Dr. Anusha Haja. Welcome back, ma'am. So I hope all of you had lunch and welcome back to our International E-Conference, Colloquium 2020. Today we are at the last day of our conference. Thanks for being with us for these 3 days. And our speaker, Dr. Anna Hajra, is with us. Before she starts, I would like to give a brief introduction about her.
Rita is also here online.
Yes, sir. So Dr. Anusha Hajra is an experienced clinical pharmacologist with a demonstrated history of working in pharmaceutical industry. Strong research professional skilled in clinical pharmacology, pharmacometrics, life sciences, CRO management, and pharmacokinetic pharmacodynamic modeling, and its application in drug development settings from early to late stage of drug development. She started her career as a research assistant in the State University of New York in the year of 2001, and she was there for more than 5 years. From November 2006 to May 2016, she holds three different positions of manager, associate director, director in Pfizer. And at first, she's holding the post of scientific director in Janssen Pharmaceuticals since January 2017. She was with the American Association of Pharmaceutical for nearly 4 years. At present, she is connected with the International Society of Pharmacokinetics and American Society of Clinical Pharmacology and Therapeutics. She completed her bachelor's in pharmacy from Dadasur University in the year of 1999. She did her master's MS from University of Toledo, and she pursued her PhD from State University of New York College at Buffalo, USA. So this was an introduction about our respective speaker. I would like to request our speaker, ma'am, if you please proceed for your presentation.
Thank you very, very much. Can you see my desktop?
Yeah, it's
Yes,
visible.
it's fully visible, ma'am.
Cool. Thank you so much again for a kind introduction. I am really honored to be back again and in the symposium forum. And I'm hoping that all of you had a great day, all day. And today is the last day, right? For the symposium?
Yes,
Is that
ma'am.
right? Okay. So hopefully
Yes,
you guys
ma'am.
had a great time. Today what I aim to do is to go through the application of a different quantitative pharmacology principle during the drug development of large molecules. So in terms of large molecule, there have been a growing trend, and as most of you may know, that as much as or more than that or equal amount of biologics have been in the clinic and is going through submission in the FDA, EMA, and all over the world. And it's dominating a growing list of more than 200 approved biotherapeutic agents in the medicine, bucket today. So if you look at the increasing number of approved biologics from 1982, you can see that it has increased, exponentially almost up to 2012. And I just took a look at the FDA website. In 2020, with all of these going on, COVID, etc., there's still number of biologics have been approved already. And in 2019, there were almost 30 plus biologics which were approved. So when I talk about biologics, they come into different categories, that is vaccines, enzymes, different blood clotting factors, etc. So today I would though focus on these targeted antibodies primarily, which are basically used as therapeutics against various antigens that are present in our body. And this could vary from various disease areas such as inflammation, cardiovascular, oncology, etc. So those are that's the particular area of biologics that I would be focusing on. So let me just see whether I can go to the speaker view. Okay. Thank you. Is there any way can you guys see this top part here as well when I'm sharing, Himanshu, or it's good?
Yes,
Okay.
it's in.
So before I just wanted to give a quick overview of what are the differences between when we call about small molecules and therapeutic monoclonal antibodies. So just from the picture, you can see that here in the corner, I put this small molecular structure of this drug called tofacitinib, which is approved for rheumatoid arthritis. So the molecular weight is about 500 Dalton. Whereas when you see in comparison, golimumab, which is a monoclonal antibody, again, used in inflammation primarily, you can see this humongous structure and complicated structure with Fabs in here, and then the FC portion. It's just like multiple fold in terms of molecular weight. So this is 150 to 151 kilodalton. So that's a big difference in terms of molecular weight, and therefore that generates a number of differences how your body treats a small molecule versus this large of a molecule. So other differences are usually the small molecules are chemically synthesized, and most of you are aware of that, whereas these large molecules are usually produced by these living host cells like CHO cells, etc. So it's a totally different type of synthetic mechanism of synthesis. And usually, as you know, like these small molecules, a single molecular entity, so it can be produced with high chemical purity and also reproducibility. And standards are pretty well established for years. Whereas these monoclonal antibodies are relatively new. They are heterogeneous in mixture, made up of various amino acids linked together. And therefore they have primary, secondary, tertiary structures. So they are extremely sensitive to manufacturing processes and then what kind of cell lines they are developed in. So the synthesis, the synthesis of this is manufacturing processes are extremely difficult to standardized, but we have come very far in terms of standardizing them. But still, at the end of the day, very different from small molecules. So there are various considerations when we develop a biologic, and which differ from small molecular drugs. So these are the list of things that usually we consider in terms of when we start developing a biologic. And I would focus on the first three points here, which is ADME differences, that is absorption, distribution, metabolism, and elimination differences between the biologics and small drugs. Then I would go into this PKPD modeling of biologics with some examples for you to see, how we can apply these modelings in order to basically recommend various doses for these antibodies. And at the end of it, end of my talk, I would just briefly go over these pharmacometric principle in pediatrics, and specifically focusing on large molecules. So what are some differences in biologics and small molecules? As I said, initially that physical characteristics, but here I'm going to go through the pharmacokinetic or the absorption, distribution, metabolism, how are they different, how do they differ from each other. So biologics are usually given by parenteral administration, such as IV or sub-Q type of administration, because in the gut, because of all these enzymes that are present, they would have extremely low stability and therefore poor oral bioavailability. So oral administration of antibodies are just non-existent. They are usually given by IV or subcutaneous route. When we give those administration, via sub-Q route, they are absorbed in the systemic circulation to go all over the body by this lymphatic systems that we have present. And usually from the sub-Q site of injection, there's very slow absorption with highest concentration in the body after can achieve in 6 to 7 days after you give the injection. So that's a very different type of absorption phenomenon from small molecules, where we give them usually in terms of tablets, capsules, so oral administration. And as soon as they are in the gut, they are absorbed through the blood capillaries in the gut. And usually the absorption from the gut is very rapid. Of course, it depends on, again, the physicochemical properties of the drug, but it can happen, like, for some drugs which are BCS class 1, that can happen within half hour to about 6 to 7 hours, but we are talking about hours versus days. In terms of how these distributes in the body, as you saw, that the size of these molecules are very, very large. So they usually tend to limit within the plasma or extracellular fluids. For example, in order to get into the CNS, it's almost impossible because of the size of these molecules. And usually these transportation within our body between the cells happens by convection. Whereas, because of the size and lipophilicity of small molecules, they tend to get into, various organs. And of course, even within small molecule, you would have differential that, some would get into the CNS versus not, or the central nervous system and not. So that's how the distribution profiles usually differ. In terms of metabolism, then how basically our body processes these two different types of drugs. In terms of large molecule, they are usually catabolized to endogenous amino acids because they are made up of the basic structure of amino acids. So proteolysis is a big part and plays a big role. Some of these, it's very interesting that how the elimination of these large molecules are, basically they're saved by these FcRn receptors, etc. So it's quite interesting, but very different than large small molecules. And because of these different target-mediated drug disposition, then you can have anti-drug antibody, all these kind of complicated system where your body is recognizing them as foreign substance. Because of that, the pharmacokinetic profiles may not be linear. What I mean by that your concentration in the body may not be proportional to the dose that you're giving. And the half-life for these go in weeks. So the recent antibody I was working on in oncology, the half-life for that drug is 20 days. So that means that in order for us to go from the highest concentration to the half of that would take almost 20 days. Whereas, the metabolism of small molecules are pretty well established. They are metabolized in the liver through the P450s are the major drug metabolizing enzyme. PK is usually linear, that there can be some Michaelis Menten type of nonlinear kinetics, but that's usually not the case. And T-half or half-life is in hours, and sometimes it could be 1 day, but definitely much shorter than large molecules. Elimination mechanism for large molecule versus small. You can see that, on the right is the small molecule. They are subject to transporter, biliary excretion, renal excretion, and hepatic metabolism. Those are different types of pathways that a small molecule gets eliminated from our body, and eventually, either through renal excretion for the total drug or the metabolized drug. That's how we get rid of the small molecules. Whereas large molecule is, there are three primary pathways. One is that your body can generate anti-MAB antibody. So that those antibody basically binds these therapeutic proteins and causes elimination of the drug. It is, of course, not very desirable when we are developing a molecular antibody to have anti-drug antibody. So that's a flag when you are developing these drugs. If you have a lot of antibody generated and that's actually causing elimination of the drug, that's not desirable. We would like to get rid of that and go for something which is not causing that kind of immunogenic reaction, which would cause elimination of these drugs. Non-specific pathway proteolysis, these are proteins, so we know that that's the way. And the other very interesting type of phenomenon for these guys are that it's target-mediated drug disposition. So when it goes to the target and in the receptor it binds, that receptor and drug complex can get internalized and basically eliminated. So we have developed models to basically characterize all of these so that we can help dosing decisions for these MABs. Also, DDI, so drug-drug interaction potentials for MABs are substantially lower than small molecules. So that gives us, sends us to the next topic, is that PKPD modeling of biologics, and what is the concept of target-mediated drug disposition that is more common in biologics. So here is a complex, interplay, of different, basically, you can imagine your body into these small compartments, and interplay of those determines the fate of the antibody. So this green is the compartment, is where a dose of the antibody is given via IV route or by subcutaneous route. So depending on the bioavailability and constant by which it absorbs from this injection site to the systemic circulation, then we have this monoclonal antibody in plasma characterized by CP, and then this volume of distribution. Now, as I said before, there could be a non-specific pathway, which is this rate constant, and then we can have some antibody in the tissue. These are the anti-drug antibody that can form. So these rate processes are going through this. This can get eliminated. So this is the pathway we talked about, anti-drug antibody that can form a complex and that can get eliminated from the body. Whereas here is the receptor. So of course, these are the MABs are directed to their receptors in order to generate efficacy, right? So there are plenty of free receptors, for example, soluble receptor, and I'll give you an example of soluble IgE, which is rampant for allergy. So for example, this MAB will bind to the receptor, free receptor, making this receptor complex. So that receptor complex can have its own elimination, or it might just be a distribution compartment. So we can measure this. Sometimes we can measure this in the peripheral compartment or even in the tissue compartment. And after we have that, we basically model how this, through various rate constants and differential equations, we would try to characterize how this drug is, the disposition of this drug in our body. So I'll show you a few phenomena. So sirocumab here, which is targeting soluble IL-6 or interleukin-6 in our body, this is pretty simple. So it seems like at the concentrations that the drug is effective, it basically is linear. That means that you increase dose and you see increase in concentration, which is the y-axis in here, and x-axis is time in day unit. So usually you would see that the pharmacokinetic profile would be highest here and then slowly goes down because of the elimination processes. And as you give higher doses, it linearly increases in concentration. Okay, there's no crazy thing involved here. Whereas if you come to tocilizumab, which is targeting IL-6 receptor, if you give very low dose, you see this like kind of funky profile, which looks like Michaelis-Menten type of kinetics. However, when you go higher in the dose, you see much similarity like this, and we say that, okay, well, at this concentration range, we have linear kinetics, so we don't have to worry about varying dose in different direction. Whereas you come here, hypothetical MAB, where you see this nonlinear type of kinetics all through the dosing. So maybe, this nonlinear binding is happening, then here some nonlinear elimination, some nonlinearity going here, giving rise to this kind of complex situation. And in this case, then, we have to be very careful about what kind of doses are we giving, because as you can see from 100 to 30, it's not like 1/3 of the concentration that we are predicting. It's much less than that. So those are the things that we have to worried about when we are recommending doses for the label for the patients. So why and how the target engagement and receptor occupancy are used in our therapeutic program. So that's something especially like, applicable for MABs, because what happens is that for small molecules, they go and binds to the receptor. That kinetics does not affect the PK of the drug. Whereas for large molecule, what happens often is that how it binds to the receptor, that kinetics actually feeds back into the PK of the molecule itself. So sometimes we do directly are able to measure the pharmacodynamic effect of the biotherapeutics by its interaction with the therapeutic target. So we can call it like target engagement when it's a soluble target, whereas receptor occupancy when it's a cell surface receptor. So characterizing this binding may not guarantee efficacy, but in most of the cases, you want to, there's a degree of desired RO or receptor occupancy that you want to achieve that would be correlated with your efficacy. So for example, I'm going to give you example of anti-PD-1 antibody. And for nivolumab, pembrolizumab, which are the leading, you want to see 70% or higher receptor occupancy for desired efficacies for these antibodies. So if one has to develop a new anti-PD-1, they would target like 70 to 100% receptor occupancy. And that's how basically RO, so receptor occupancy is driven by PK, and PD, so that efficacy is driven by RO. So that's how we also use RO or determination of RO in the therapeutic programs. So here, I just wanted to give you an example where modeling and simulations were directly applied to, and then this happens for many programs, therapeutic programs in the current years. But this particular antibody, omalizumab, which targets soluble IgE, which is indicated for severe persistent asthma or chronic idiopathic urticaria. This is a hallmark of how modeling was applied to directly, recommend the doses in the various like complicated dosing regimen in the label. So this humanized anti-IgG molecular mAb binds and captures the circulating IgE, preventing this IgE to interact with the receptors and mast cells. And that's how it interrupts the allergic cascade. And that's how it is like, by mechanism of action, it is indicated for asthma. Now the mechanism-based model of omalizumab suggested that decreased IgE production in patients with IgE-mediated asthma upon long-term treatment. So essentially, this free omalizumab is binding to the circulating IgE, but indirectly, it's also reducing the IgE production. So over long term, like I'll show you some figures, over long term, you can see that, if somebody is on long-term treatment, they don't need to be on the treatment forever. It's actually reducing the synthesis of IgE, and that's how it's preventing the disease. So pretty complicated figure, which I got from this particular literature or the paper that I gave the reference to here. Two extremely different, like, so this female, this is the woman with a different body weight and of course gender, and here, and she comes with a different IgE level baseline, whereas this is a male subject with a different IgE level and weight. And when this omalizumab, so the green curve is the monoclonal antibody concentration, and here is the, as that goes up, it binds to the free IgE, so it goes down like that. And then these are the observed total IgE. And you can see that the solid line, which is the model predicted concentration, they line up with the concentration as well as free IgE, as well as the IgE production. So what the authors basically conclude from here is that if you measure the total IgE in the blood, that is pretty consistent with the IgE production. And these guys, over time, you see that the IgE production is going down. Whereas this curve is shallow for this individual. However, this is going down. So what they're trying to say is that this model is able to accommodate various weight range, various baseline IgE, and almost will give you an individualized regimen for each individual. So based on their body weight, age, and baseline IgE, and over time IgE, you should be able to tell what doses would be given for these patients. There are some other type of predictions they make that if you give the the drug for 1 year versus 3 years versus 5 years, you see that prolonged decrease of IgE production. So that basically helps understanding that how this drug works and how long you have to give treatment. And just wanted to give you like an example how all of this ended up in the label of the drug, right? So you can see that the recommendation is various body weight category, here baseline treatments, and you just go like, so if somebody is within this 60 to 70 kg with the baseline of this, they would take 300 milligrams of omalizumab. So pretty cool, you just use all those kind of rate limiting stages and then eventually come to the dosing, and that's what matters for the patients and the physicians, right? So the last part is that, how do we use the pharmacometric principles in this, precious special population in pediatrics? So it is a drug developer's dilemma that, the therapeutic dogma is that it used to be, okay, children are half adults, so let's give them half the drugs. That's not quite true. So physiological perspective, we have various practical questions like how early can the adult experience be applied to children? So we have all these drug development experience in adults, when can we, at what age we can apply that to children? And this goes beyond like, large molecule. We have to think all of that for small molecule drug development as well. Can adult model be scaled down to children based on body weight? So whatever we are giving, for example, 70 milligrams in 70 kg, if the child is 1 kg, should we be given 1 mg? Like, can that be done? So something like that, like, a 70 kg 10-year-old, is that child equivalent to 70 kg adult? Is all the systems that, processes the drug in the body, is it matured enough? So that's another question. As 20 kg or 1 old is equivalent to infant or not, same weight. So, and then what about therapeutic protein? So how is it different from small molecule? So all of these questions we have to think through when we go for pediatric drug development. So what is the situation for development of therapeutic protein in pediatrics? Now, because they are very targeted, so the systemic effect is much lower. So therefore, it can be very attractive for pediatric development because the side effects might be much lower. However, a lot of research and effort have been made to understand the ontogeny of CYP3A, for example, metabolizing enzymes for drugs, and how that process develops in pediatrics from infancy to 10 years, 12 years old. However, this process for therapeutic protein is pretty limited. Like, it's a very young type of field in drug development. And then, of course, there's a lack of consensus. It's a very, young field of research. So there's a lot of differences in opinion how these therapeutic proteins should be dosed in kids. So again, ADME considerations in pediatric dosing. We need to understand the age and size related changes in drug disposition for efficacy and safety, then patient-specific factor, whether the subcutaneous tissue in children, whether it has same lymphatic system or not. In adults, that would determine the absolute bioavailability or the absorption process. We are going back to the ADME again, and how that is applying to children. Then propensity of anti-drug antibody development, because that would determine what would be the clearance mechanism from the body. So for some higher ADA incidences reported for adalimumab, abatacept, whereas for some ADA, the reports are lower in children. So drug by drug basis, you need to figure out how the children are reacting to those versus adults. So here is the golden Snail rule, basically, that golden curve, that even as complicated a structure as the snail shell is supposed to be, like, there is a method behind the madness, and every angle, like, as the snail grows and the shell goes, that can be actually deciphered by mathematical modeling and these angles. So for our body, as complex as it sounds and looks and etc., we have seen that most of the body size relations to the pharmacokinetic parameters can be simplified into this simple equation, which is a power equation. So for example, if we say that it's clearance, that what is the rate of this drug, the rate by which this drug is being cleared from the body, that is actually quite correlated with your body weight, surprisingly. And this coefficient, if you can characterize them, you can actually tell that with body weight differences, how the drug will act in your body. So that's pretty cool that such a complex system can be drawn into this the apparently simple formula. So having said that, I would just go through a couple of few different types of methods that we use when we dose pediatrics. So one and foremost, the most popular one is the mg per kg basis. For example, you have 5 milligrams per kg infliximab in pediatric ulcerative colitis or pediatric CD. So there is no issues like, the child comes, the doctor weighs the patient, and okay, fine, 10 kg, so you need 50 milligrams of it. Very simple, right? Then of course, I will just discuss a few issues with that. So then the second is the BSA basis, body surface area. Adalimumab is also given or is given based on per meter square or BSA based. Tiered fixed dosing is that, you say that, okay, over 35 kg, you have a drug, you have this dose, and then body weight at less than 35, you have a lower dose, something like that. And then hybrid would be like in that lower body weight, you just go through this mg per kg, but after a certain period or after a certain age and body weight, you can just give a flat dose. And etanercept is something, it's a RA drug that's given using this principle. So how effective is body weight correction? So it's very simple, as I mentioned, and achievable, exposure in older children for 10 years and older. It works very good. However, what happens is that one of the weaknesses in very young subjects, it can, produce like underdosing. So we need to do some, adjustment for lower body weight subjects. And modified, here again, it's showing that you can modify this to do like, it's less than 30 kg or over 30 kg. So I'll show you what happens here, that as a child grows through the age here in the 1 to 18, you can see that after age of 11, here is the body weight corrected clearance that is flat. So therefore you can give some dose like this, that mg per kg, and that should take care of it. But in lower body age children, your clearance is higher than here. So you have to adjust for that high clearance. You have to give a little bit higher dose in terms of mg per kg to compensate for that higher clearance. And infliximab does that actually, or sorry, body weight correction seems to work in that, and this work was done in infliximab to demonstrate that. Now, body weight, body surface area is physiologically more plausible. It achieves comparable systemic exposure over wide body range. It doesn't have that issue. Like, you can see that if you, the BSA corrected clearance is flat over the age range for here. And this is clearance and volume of distribution. It works for both. But the weakness is prone to dosing error, like, to calculate, like, one kid is coming to the clinic and getting their body surface area, the measurement might be wrong. So that leads to dosing error. But this particular method works actually pretty well for children. And then tiered dosing is that, as we talked about, that less than 35 kg. So that takes care of kind of both worlds. Body kg, like per kg and per BSA. And the weakness, however, is that may not provide very uniform systemic exposure throughout the body weight range. So if you do that, you might have this concentration and then going up all of a sudden. So that kind of a little bit different, but it's indeed another way to do that. And then this hybrid dosing, it is relatively simple. It avoids overdosing in subjects with high body weight. So you can see that, this etanercept clearance increases and then it flattens out. So what we can do is up to this body weight, we can say that, hey, let's go with, 63 kg or more would get some mg per kg dose. And after that, you can just give, when it plateaus, then you can give something else. And just wanted to demonstrate that these days, modeling simulation is playing a big role sometimes, that the dosing regimens we don't even study by modeling and simulation using all data that's the prior data, you can actually have labeling for the doses that have not been studied. So for given on the basis of simulation analysis, the USA Food and Drug Administration approved in so etanercept, they basically, the change of dosing regimen was approved based on modeling simulations only and without generating data. So that's really good. So for pediatric development, we still have more questions than answers, because a lot of these, as we talked about, that absorption, how the lymphatic fluid drainage in pediatrics is very limited. Therefore, it's almost case by case basis. We have to do thorough analysis and investigation in pediatrics to determine that distribution. Again, we don't know blood and plasma volume per kg body weight versus age. That might change over time. So more needs to be investigated in that. And also in terms of, how this FcRn density, avidity, how the TMDD works in children versus adults, how ADA is like in children versus adults. Those are the things that are unknown. So there are lots of, therefore, scope of application of our quantitative pharmacology. We can use, all our knowledge to go with the starting dose, and through small trials, we can gather more data and further refine our models to propose various doses. So our recommendation for any throughout the drug development process, especially for pediatrics, that we need to drive defensively using pharmacology as a guide. So this was the last slide that I had. And with that, I would take any questions or comments that you may have. Thank you. Thanks for your attention.
Thank you, ma'am. If anyone has any question, they can write it down in the chat and ask
Yeah.
ma'am. Ma'am, I have a question for you.
Sure.
Ma'am, we have seen, or we do see, that there are many common side effects of the MAB treatment. So with your treatment protocol, will the side effects reduce? Can we see that happening?
Yeah, so in the sense, it's not a different, paradigm. So so side effects, for example, you would have for infusion-related reaction, right? Or injection site reaction. So definitely, if those are, we can measure them and model them, then using application of modeling simulation strategy, you can optimize that side effects to be lower. For example, there is this, and it applies for both small molecules and large molecules. There was this one particular drug for which we were getting a lot of thrombocytopenia as a side effect, right? So, and the efficacy, we were able to actually characterize how the thrombocytopenia profile is with concentration. So we were able to, come to a dosing regimen, which will be right in the middle. So your efficacy increases over time. Your thrombocytopenia, your thrombocytopenia would increase with time, and then your efficacy will plateau over a dose regimen. So you need to get that exactly optimum dose to minimize the side effects and maximize your benefit. So that, yeah.
Thank you, ma'am. Ma'am, I have another question. So I'm going to read it out for you. It's written here. Can MABs be developed against the ACE1 binding site paratope of COVID-19?
So I'm not very knowledgeable in the COVID area. I have to acknowledge that. But having said that, actually, you know what? I do not know whether for that particular binding site or I don't have knowledge in that. I just know that there are maps being developed, but I haven't looked into details in terms of COVID.
Okay. Thank you so much, ma'am.
Yeah, of course. You guys have the rest. Is it the rest of the good time, productive time?
Yes.
Thank you very much
Thank you
for this
so much.
for this opportunity.
Thank you, ma'am. Ma'am, we have a small token of appreciation for you. I would request Sakshar to kindly show it to ma'am, because we are in a virtual world now. So,
Yeah.
Yes.
Sakshar?
Hello.
Yes, you're audible.
Yeah, thank you. Yeah, ma'am, wait a bit. Let me know if my screen is visible.
Sure. Yes.
Ma'am, this is what we have for you.
Ma'am, this is for you.
Thank you so much. Thank you. I appreciate it. I appreciate this opportunity.
You're welcome, ma'am.
Thank you. Thank you. Thank you. Okay.
So moving on to our next session of the day, we have the last plenary session of our day 3 of International E-Conference. Dr. Mukesh Nandave. He is an associate professor in the Department of Pharmacology at Delhi Pharmaceutical Sciences and Research University. Dr. Nandave earned his PhD in pharmacology from AIIMS Delhi and received his postdoctoral training from the Division of Cardiothoracic Surgery, the Ohio State University Medical Center, Columbus, USA. Since more than 12 years, Dr. Nandave has been investigating the role of nutraceuticals, herbo-mineral formulations, plant extracts, and constituents for myocardial ischemia and reperfusion injury, diabetes, obesity, pain management, etc. He has published more than 65 papers in peer-reviewed national and international journals. His overall citations are now at 761 with an HI index of 17. His lab received more than 1.5 crore total funding from the government as well as industry, including Dabur, Madhav, Bor, Sandhu, etc. Dr. Nandave has received numerous awards, including the D. Achari Gold Medal by Indian Pharmacological Society, Association of Pharmaceutical Teachers of India, APTE, Young Pharmacy Teacher of the Year Award, and so on. We have Dr. Nandave with us. So, sir, the stage is all yours. You may proceed with your presentation, sir.
Yeah, 1 second, Prita. 1 second, Prita. Hold on.
Yes, sir.
Yeah.
Am I audible?
Nandave, yeah, you can speak. Yeah.
Okay. So, good evening to all present here, and I'm really thankful to all these speakers and organizers. First of all, my simple expectation from this all the audience is I don't know. I also listen in a double voice.
Sir, you can send me the PPT. I can share on behalf of you.
Okay. Is that possible?
Yeah, in WhatsApp also, if you can send me, I'll do that.
Okay. I'm accessible on email actually.
Yeah, you send it to my mail, sir. It's HSMAJI
HSMAJI
@ JISUniversity.ac.in.
AC.in.
Yeah.
Okay. Should I continue?
It's not yet reached in my email, sir.
Maybe I have some videos actually, so I'm okay with.
Okay, sir. Okay, sir, you can continue.
Okay. Yeah. So My topic is driving innovations to defeat diabetes. And why I chose this, because it's very close to my heart. I have been working since last more than 10 years, and I personally found it's really interesting to work in diabetes, understanding day-to-day problems of diabetic patients. Now, what are the simple expectations of this presentation, or maybe you can say objectives or overview of my presentation, is to update you on innovative solutions. Basically, through this presentation, I want to focus on what are the innovative solutions available to measure the glucose level, and that's continuously, not by fingertip, and to deliver various antidiabetic drugs, including insulin, through various modes. Then artificial pancreas, what is the status of diabetes vaccine, and cell generation therapies. Now, before I talk about the innovation, what is my personal experience to manage the diabetes? In fact, I would like to quote a scientist who worked throughout his life on diabetes and found that it's a basic triangle. And even though research is there, or maybe the patient is there, the person has to focus on that basic triangle, that is physical activity, then prescribed medicines, and diet. So when person can develop a coordination between these three major of vertical things, then still that person can manage the diabetes very well. Now, you already know the recent numbers. There's 422 million adults have diabetes.
হ্যাঁ এটা কি করব, স্পট করব, স্পিকার ভিউ করে দেব, এখন দেখা যাচ্ছে? আমি অল ভিডিও
Should I
প্ল্যাটফর্ম
continue? Hello.
Yes, sir. You should continue. Yes, sir.
Yeah.
Sure, sure.
You have a big number in case of diabetes, and simply to tell that one person in 11 people is diabetic, and this number is going to be increased. Especially for India, it's more important because we are second in the number of diabetic patients. So the first thing, that is continuous glucose monitoring. Now, this monitoring is more than important than the base treatment the patient is taking, because continuous glucose monitoring help you to monitor or observe your disease very closely. And when you monitor something very closely, you can understand it and you can manage it in a better manner. So what we have, the fingertip blood testing, which is very painful, inconvenient, and there are so many limitations. But then there are technologies that are already in the market, and they're very convenient and very effective in monitoring the diabetes on day-to-day basis. So what you have, based three CGMs, you can say continuous glucose monitoring devices. The first one is Abbott's Freestyle Libre, then Dexcom, and then Medtronic. So if you see, Freestyle Libre from the Abbott is what you call very simple, cost-effective. This has only a thin fiber that goes into subcutaneously in the body and measures blood glucose for almost 2 weeks. Continuously, every 15 minutes, the blood sugar is recorded. Limitation of this freestyle is, once you apply this sensor, you cannot know your glucose level on daily basis. You can only what you call know the readings at the end of maybe 2 weeks. But what is help or advantage of this is very cost effective compared to the other CGMs. Now, uh, where it is particularly used, when the patient is newly diagnosed and the insulin dose has to be calibrated, then uh, diabetologists can use this freestyle and calibrate the dose. Then next comes J6 is another best CGM, and advantage is that it gives a sound alarm for higher low glucose, and maybe less than 70 milligram per deciliter is considered as a hypo, and more than 150 is a higher for diabetic person. Now, in terms of accuracy, Dextrom is best. Then, in terms of automatic glucose data sharing, both Dextrom and Medtronic's CGM is very effective. Then, Medtronic's Guardian is what you call advantages in terms of automatic glucose data sharing. Then, this smart diabetes software or app is already available. Then predictive technology or predictive software is in Medtronics only. Means it will tell you in advance the sugar will go up or down, so that patient can take the corrective action in advance. So these technologies, I already mentioned that what is available in the market and used in a clinical practice, but there are a few innovations which are either in a developed stage or in a pipeline or in a evaluation stage. So GlucoTrack is another option from Integrity Application Company that measures the glucose using electromagnetic waves. Now, major advantage is that all three previously listed CGMs has to be what we call injected. Means you have to prick the needle, and then only they will give the continuous glucose readings. But this GlucoTrack is a non-invasive that measures the glucose on the basis of electromagnetic waves, and this is already available in Europe. Then GlucoSense is another technology that measures the blood glucose using laser light, and Mediwise is another company that use the radio waves. A graphene patch from University of Bath that having greater accuracy because of graphene presence, and sugar level are individually measured in multiple hair follicles. So that is the reason it gives a better accuracy. Now, what is the major limitation of these technologies? The accuracy. The listed three CGMs which are available clinically, they are not that much accurate, but yes, the reproducibility is there. For this GlucoTrack, GlucoSense, and Mediwise or graphene patch, they have a limitation of accuracy. Now, you might have heard about the smart contact lenses from Google, and it's basically very tiny contact lens that continuously sense the glucose in lacrimal secretion, that is tears. So I will use very non-technical layman's term because there are many people join through my Conquer Diabetes blog as well as a Facebook page. So in between, I will simplify for them. Now, another needle-free revolution because of this innovation of non-invasive techniques to measure the blood sugar. So Know Your Science, a company from Netherlands, is going for a tiny device that can be placed under eyelid, and it's more affordable because it's very tiny and it's very accurate, and it can be this device can give the very continuous glucose readings. Uh, you can use the monitor to take the record and share them with the physician. Now, what are the various anti-diabetic drugs and how they are delivered in an innovative manner? So the first thing, the drug, earlier concept that one drug, one target, and one disease. But now there are drugs which are used in more than many diseases. There are the drugs which act simultaneously on multiple targets, multiple organs to give a better effect. So one of the example is imiglimin from Oxford company, and this drug is simultaneously particularly effective on pancreas, liver, and muscle. In pancreas, it what you call increases the insulin secretion. In liver, it acts what you call against the glucose production, and in muscles, it improves the glucose uptake and utilization. So, by increasing the insulin secretion, it produces the hypoglycemic effect and it takes the control of glucose. And in liver, uh, what you call it increase the decrease the glucose production, and in muscles it increases the glucose uptake. At present, it is in phase 3 trial in Japan, and the company is planning to launch in or evaluate in Europe and other regions. So this is the innovative way that the drug is targeting in a simultaneously in different organs and different targets and producing one common effect, that is decrease in blood sugar. Now another that is GLP-1 analog, that is semaglutide, that's from Novo Nordisk. Before the approval from the FDA, this GLP-1 analog has to be injected once in a week. But this orally available GLP analog, that is semaglutide, can be used once or twice a day, and it made the patient injection free or the needle free. So this is another advantage of innovation or research continuously in anti-diabetic therapy or care. Now this Intarsia, uh, it measures the consistent and continuous vertical delivery of exenatide via osmotic mini pump. So basically, if you see, it's a small matchstick size device can be implanted subcutaneously, and the same drug can be used for a year. So when you're minimizing the dosing frequency, when you're minimizing the injections, then the automatically the patient compliance will definitely improve. So this is a simple structure that how this Intarsia works, and this continuous delivery at a small dose is successful in completing this blood sugar level in patients. Now, this is again in an advanced stage. In fact, it is really a particular advantage for patients. Now, you have so many insulin pens. I remember that the particular broad gauge needles and then storage of insulin, then taking the insulin from the vials was very inconvenient for the patient. But now these smart pens and use of needles 32 gauge is really least painful. Now, what is specialty about this smart insulin delivery in pain? Uh, it delivers even in a very small dose, half unit doses. It's compatible with the most commonly used insulin types in market, monitors insulin temperature, and its data log can be what you call shared on a smartphone via Bluetooth, and it lasts for whole year. Okay, so you don't have to change the pen frequently. Now another advantage, at present, whatever the commonly used pens, the major problem faced by the patient is that patient usually forget to take the insulin, or the patient cannot recall that when he or she took the last dose. Now, hello? Okay. So can you hear Dr. Maji?
Yeah.
Yes, yes. Yes, sir.
So this pen is really convenient, and it saves your 16. See, hello.
Yeah, it's okay, sir.
Okay.
Actually, both the systems, the microphone is on. That's why creating a huge problem. You are speaking from the mobile phone, sir, or from the laptop?
No, I'm speaking from headphone only, the mobile.
Headphone, mobile. Yeah, you can continue, sir. It's okay. No, it's okay.
In fact, I muted my laptop mic.
No, the receiver is not muted. Okay, sir. Please carry on.
Okay.
And I haven't received your email till now, so that I could have shared. Yeah.
So this is what the smart pen. Now, artificial pancreas or closed loop system. Now, artificial pancreas and closed loop system, are they same? Yes. But why closed loop system? Because this traditional system, how this insulin is used and how blood glucose is monitored, that patient measures the blood glucose by a fingertip method. Then whatever the blood glucose level is what you call used to decide the dose. Now, this dose is decided based on what is the blood glucose level. Now, this in a closed loop system, you have three major components: CGM, then what you call insulin pump. Now, this CGM continuously measure the blood glucose and what you call sends the glucose level continuously to the insulin pump. Now, in insulin pump, there is a what you call algorithm that calculates the glucose level and accordingly decides the dose and delivers subcutaneously. Now, this is simple diagram to tell what is the closed loop system. Now, if you see, you have the continuous glucose sensor that sends the what you call data to the insulin pump, and you have the memory here or the brain here, which process this information and calculate the insulin dose and delivers through this cannula subcutaneously. And again, this action of insulin decreases the glucose level, and this is again sensed by the continuous glucose sensor, and this accordingly the signal is sent to the insulin pump. Now, how it is what you call working. Let me show you through the video.
Hmm.
So if we see, this is the implant, means vertical application of this CGM, and then pocket size black in color is a pump. This is the pod that contains the insulin reservoir, and this is the first artificial pancreas vertical approved by FDA, and more important, only the patient more than 8 year old. So this is the artificial pancreas, and this actually meaning of closed loop system. Now, at present, you have three most used or effective uh, artificial pancreas or insulin pumps available. The first and most used or advancedly developed or innovated is the Minimade 670G from Medtronics. The specific thing about it is automatically adjusts the basal insulin. And why it is? Because it is linked with the CGM. So CGM sends the data to the insulin pump, and insulin pump decides the dose and calculates the basal as well as the bolus dose of the insulin and injects this. The limitation of this, the display panel, when you have to always clip this insulin pump. Whenever you want to see the reading, you have to unclip and then again see the display and then decide the dose. Now, Omnipod Dash is the only tubeless insulin pump on the market available. Now, this Omnipod is smallest tubeless, and that's why it is very convenient for patient during the physical activity or maybe the rigorous exercise. Now, T-Slim is the only smallest and thinnest insulin pump available in the market. Then, what is the diabetes vaccine status? You have Vax, that's the French company, is developing the vaccine for type 1 diabetes. And this is basically based on quinoid vaccines. So they have their patented technology, and this vaccine, quinoid-based vaccine, is more advanced for the lupus erythematosus, and the same technology, same platform, that clinically evaluating for the type 1 diabetes, because type 1 diabetes is like lupus, autoimmune disorder. This quinoid vaccine stimulates the immune system to lower the level of inflammatory proteins, and this inflammatory protein decrease leads to the decrease in autoimmune response, and that's why the beta cells are spared from that immune attack. Now, if you see another advancement, that is cell generation therapies. Now, cell generation therapy, there are so many players working in this direction, but then their common way of working is they are working either at a site or to sustain or to supply. What it means that whenever you implant or transplant this beta cells, the immune response vertical is triggered, and then again the transplanted beta cells are killed by our immune cells. Now, what they are doing, they are engineering a home for beta cell islets. Now, what is that engineering a home for islets? Means they implant it in such manner that they are protected from the immune attack. Then sustain means ensuring the islet cell survival. So again, providing a customized environment to the implanted cells so that they are what you call preserved, protected, and allowed to multiply, proliferate. And supply is creating more insulin producing cells. So this is what 3S: site, sustain, and supply. Now, the major advancement on 3S is done by BioHub. This is basically a bioengineered mini organ where insulin producing cells are encapsulated within a protective barrier. And this leads to the long-term islet transplant recipients show near normal glucose control. And this is the phase 3 trial outcomes. And last October, they declared their results of phase 3 trial. So very soon, they will have some technology that can be used to transplant the beta cells and protect them from the immune attack. Now, another technology from wire side, the PEC Direct, PEC NCAP, and PEC QT. So PEC Direct is specifically for type 1 diabetic patient who is having the highest risk of life-threatening acute complication. PEC NCAP is indicated for potential function cure for type 1 diabetic patient. And then PSCQT is a next generation therapy for all insulin required diabetes. Now, PSCQT basically uh, specialized cells, they are implanted and which are resistant to the death caused by immune attack. So that's a three-way. This YSIT company trying to replace the cells, beta cells, and reducing the requirement of insulin. Now, organogenesis was received the FDA orphan drug designation for autologous insulin producing cells in treatment of severe hypoglycemia prone diabetes. That is particularly resulted from total pancreatomy. Now, if you see, this is another advantage that when you implant uh, the beta cells of other patient, then rejection is highest possibility. But the autologous insulin means producing cells means you isolate beta cells from the same patient and use for the same patient. So acceptance is more. Now, Insights is another company which is working in immunotherapeutics. They are successful for developing this MCY 0098 used in type 1 diabetes, and they are in a human phase 1B trial. Now, what is Insights is basically again a technology. Now, if you see this video,
epitopes are peptides made of a thioreductase motif linked to an epitope derived from an autoantigen targeted by the autoimmune response. Upon injection into the body, case of type 1 diabetes, in the pancreas, insulin-producing beta cells are destroyed by pathogenic T cells. Epitopes, a technology developed by Insights, have the potential to block these deleterious immune responses. Epitopes are peptides made of a thioreductase motif linked to an epitope derived from an autoantigen targeted by the autoimmune response. Upon injection of the response, the epitope is presented at the surface of the antigen-presenting cell. When epitopes are presented by the antigen-presenting cell to naive CD4 T cells in the lymph nodes, the thioreductase motif will direct the maturation of the CD4 T cells into cytoplasm.
Just to explain here, what they have done it,
The cytolytic CD4 T cells migrate.
so
Dr.
what they have
Nandave,
done it, yes,
sir, we are running out of time.
okay, I'm done.
Sure, sir.
So this technology is basically prevent the immune attack, and this is another advancement, microchips, basically in the form of nanorobots that help to diagnose the diabetes, type 1 diabetes in advance. And these are the some mandatory screening requirements. Like once this diabetes happens, the person has to manage only. But if you can detect in advance and at an early stage, you can use the immunotherapy or various approaches so that disease cannot progress at an advanced stage. Now, another thing, even though there are so many advantages or innovations, but still we have to wait for the complete cure. And that's why we have to remember that we have to focus on this basic triangle, that is coordination between the diet, medicine, and physical activity. Now, so this is what you call unbelievable or maybe very optimistic future, but yes, it's possible as far as whole human being or community works together to find out the cure. And this is all from my side, and I'm really sorry for this technical problem. In the last moment, my video camera is not working. So you can reach me through my Gmail, through my blog and Facebook page, Conquer Diabetes. Once again, thank you very much to all the organizers and especially Dr. Machi for giving this opportunity. And
thank you, sir. Thank you, sir. Rita, I think we can go for question answer session so that we can see, we can see share on the screen.
Yes, sir.
Sir,
So we have a couple of questions for you.
Sir, please put your video on and
yes, sir.
So that we can see you, sir. People can see you. Yeah. Oh, Rita.
Sir, Quillen sir is also here.
Yeah. Sorry.
Oh, wow.
We will have question answer session.
Sure.
Let me talk to Dr. Carlin first. Let me unmute him.
Boss.
Hello, Dr. Carlin.
Good morning. How are you?
Yeah, I'm fine, Dr. Carlin.
Although I guess
yeah.
Evening for you.
Yeah, it's evening. We are just ending the session, and after this only we'll start the valedictory. Please stay with us, sir.
Okay.
Yeah.
Dr. Nanda, we have a couple of questions for you in the chat box. I'm going to read it out for you. So the first question would be like, for the eye pen which you were mentioning, which is used for diabetes, what is the exact mechanism which is being used in the eye pen? Is it something like reverse iontophoresis or is it something else?
In eye pen, it's not actually way of delivery. It's basically they have used the thermal sensor. They have used this engineering basically that records the data log, like how much dose was taken by the patient last time, and it has a timer actually. In other words, this iPen is also called as uh, time insulin. Time insulin basically tells what time that patient took the last dose and how much was the dose. So when you uncap the pen, that moment or that moment is recorded in a software, and that is what you call provide. And these 16 readings are recorded so that last 16 doses, their timing is given to the patient.
Thank you, sir. So another question which has come up. Sir, the insulin pumps which you were mentioning, are they affordable and available in the Indian market?
Yes, they are available. In fact, Medtronics have three versions available. The basic version is only a delivery actually. It's not linked with any CGM. So whatever the insulin patient takes by maybe syringe or by pen, it is delivered by the pump. Now, what is the advantage? Patient has to take minimum 3 to 4 injections every day when he or she use the pen or syringe. Now, this insulin pump gives you advantage that every 3 or 4 days you can change the insulin cartridge, and you have to only subcutaneously implant that injections at the injection site. Now, in the long term, what is the advantage of the insulin pump? That insulin sites become functional and remains functional because of less insulin injections. Then another advantage, the accurate dose. So when the zero point 0.5 or 1 unit matters a lot, roughly 1 unit reduces the 50 to 60 milligram per deciliter sugar, blood sugar. Now imagine when a person take the inaccurate dose, then obviously the person will have abnormal high or low sugars. So this is basic pump available at maybe 1.5 lakh. And then the most advanced, what I showed, 670G, is available for 5.5 lakh. That comes with the closed loop system. There is a CGM, there is a insulin pump. Now, this will help you to control the blood sugar nearly normal.
Thank you so much, sir. So we are getting many more questions, which we'll be mailing to you since we are a little short on time today. So we are really thankful that you came to our international conference here. Thank you so much, sir, for coming. Sir, we have a small token of appreciation for you. I would request Saksha to kindly show it to sir since we are in a virtual world now.
Yeah, I agree.
Sure, just give me a moment.
Yes.
Let me know when my screen is visible.
Sure. Sir, this is for you.
Yeah, thank you so much.
From us.
And I really apologize for this technical glitch.
Thank you, sir. Thank you so much.
Thank you, sir. Thank you, sir. Yes, sir. Thank you.
So with this, we come to the end of our plenary sessions of today.
Sir, I request Dr. Mukesh Nandave and Carlin, sir, to stay back with us in our valedictory session also,
Yes,
sir.
sure.
Yeah, thank you. It's over to Preeta now. just give me 1 or 2 minutes.